These are some important simple symbols representing the enzyme, the substrate, the enzyme substrate complex, the enzyme product complex, and the product. Okay, so here it is. We have the enzyme plus the substrate, and it goes to the enzyme substrate complex. And then in between the ES and the EP, that's actually where the reaction takes place. So that second arrow is where the reaction occurs. So then we produce products. So EP stands for the enzyme product complex. And then that next arrow, the product leaves. And then the enzyme is by itself again and able to be reused for the next substrate. In order for that reaction, in order for that reaction to take place, the enzyme and substrate must come together. The substrate goes into the active site of the enzyme, and the weak forces are holding them together. Because the substrate's not going to stay there, we saw that after the reaction, the substrate leaves, or the product leaves, and then the enzyme is able to form interactions with other substrates. Two different models describe how the enzyme and the substrate bind together. And again, that's a temporarily binds to the enzyme. But first is the lock and key model, where a specific key is going to fit a lock. So the substrate matches the active site exactly, just like a key fits, or an exact key fits a lock. The next model is the induced fit model, where the substrate binds with the enzyme, and it's not a perfect fit like the lock and key model. It's a loose spatial fit. So you can see there's a little bit of room between the sides of the enzyme and the substrate. So the enzyme will undergo a conformational change that will allow for a tighter fit. And so after that occurs, then the reaction takes place. Proximity effect describes the movement of the substrate molecules when binding to the enzyme's active site. So the enzyme guides and steers the substrate into the active site. Then the orientation effect, the enzyme guides and steers the substrate into the active site, but it does it in a specific orientation to make the reaction occur. So these enzymes and these substrates are moving around and running into each other, but the enzyme's able to steer and guide that substrate in to the active site exactly how it's supposed to be. Then with the catalytic effect, are catalytic strategies to push the reaction along. So here's just some examples of that. We have the acid-base catalysis, which will move protons. There's covalent catalysis that has an electron carrier, and electrostatic catalysis, it stabilizes charges. So again, those are just some examples of the catalytic effect. The energy effect, the enzyme induces structural rearrangement, which strain the substrate that's bound into a position closer to the transition state, and this will lower the energy difference between the substrate and the transition state, and it helps catalyze the reaction. So these are just different enzyme mechanisms that occur between the enzyme and the substrate. There are 4 factors that will affect enzyme activity. We'll look at substrate concentration, enzyme concentration, temperature, and pH. So first is substrate concentration. So the higher the substrate concentration, the faster the enzyme will work. So until all available enzyme active sites are being used to catalyze reactions. That's why the graph plateaus. So it increases, so you can see that the y-axis is reaction rate, the x-axis is the substrate concentration. So as the substrate concentration increases, the reaction rate increases. But once all those enzymes have a substrate, that reaction rate plateaus. This is called the saturation point, because all the enzymes have a substrate and the reaction rate is working at its maximum capacity. So where it plateaus right there is the saturation point. Also, enzyme concentration affect the enzyme activity. So enzyme concentration increases with by increasing the enzyme concentration, the maximum reaction rate greatly increases. So you can see on the graph, the y-axis is the reaction rate, the enzyme concentration is the x-axis, and as that enzyme concentration goes up, the rate of the reaction goes up as well. As long as there's enough substrate to bind to. So that's why underneath enzyme concentration it says excess substrate. So there has to be enough substrate for the enzyme to bind to and cause the reaction. Temperature also affects enzyme activity. So raising the temperature will speed up the reaction. Activity will then decrease at a lower temperature. So increasing the temperature increases the rate of reaction, but eventually we reach a temperature where the enzyme will become denatured. And then the reaction rate abruptly declines. So that's why we have an increase and then a decrease, because at those higher temperatures, the enzyme can become denatured. So just to point out, the enzyme rate increases as temperature increases because molecules, their speed increases when we have an increase in temperature, and so we have more enzyme substrate collisions, and they're able to come together and make that reaction happen. Okay, then pH has a significant effect on enzymes. It can affect the intramolecular forces and change the enzyme's shape. So enzymes have a pH range in which they perform optimally. So most enzymes have their maximum activity between a pH of 5 to 9. So you can see that in the graph. With trypsin, it has a pH range from 5 to, well, a little further than 9, probably 11. But then pepsin's pH range is lower. It's more acidic. So, depending on what enzyme we're looking at will depend on where their optimal pH is. We're going to skip this slide. We've already talked about it, so we're going to skip this one. Okay, so enzyme activation is any process that initiates or increases the action of the enzyme. And then really what we're going to focus on right now is enzyme inhibition. It's any process that slows or stops the action of an enzyme. So we kind of looked at enzyme activation just a minute ago with increasing temperature, increasing substrate, increasing enzyme. Also the cofactors we've talked about. So really we're going to focus on enzyme inhibition right now, where it slows or stops the action of the enzyme. So an irreversible inhibitor will bind to an enzyme active site so that a substrate will not be able to enter the active site. So it will covalently bind to the protein so the enzyme becomes denatured. So poisons and heavy metal ions are an example of an irreversible inhibitor. A reversible competitive inhibitor will bind non-covalently, so it's a little less The binding is not as stable as the irreversible inhibitors are, but it will bind non-covalently to the active site, but no reaction takes place when there's a competitive inhibitor in the active site. So this will prevent the substrate from binding to the active site. So we can see that here. You can see the competitive inhibitor is in the active site, it's bound to the active site, and the substrate is not able to enter. So the inhibitor has a similar structure as the substrate, and it can fit into the active site and prevent that substrate from entering and binding with the active site. A non-competitive inhibitor does not bind to the active site, but other parts of the enzyme. So when it does this, it can change the conformation of the active site, and that would prevent the substrate from entering. Or an uncompetitive inhibitor binds to the enzyme substrate complex. So you can see on that picture, we have the enzyme and the substrate. They've already binded together, but then we have the uncompetitive inhibitor binding, and it causes the reaction to occur less efficiently or not at all even. Okay, covalent modifications are enzyme-catalyzed alterations of synthesized proteins, and they include the addition or the removal of chemical groups. So some enzymes are synthesized in an inactive form. These are called zymogens, or proenzymes. So basically, they're inactive precursors of enzymes, and it requires a biochemical change for it to become an active enzyme. So here's an example of that. We have chymotrypsinogen, and it's an inactive enzyme, and it's a precursor to chymotrypsin. So chymotrypsinogen reaches it has to be inactive until it reaches the digestive tract, and then there it will be activated by trypsin. So trypsin will cleave the peptide bond between arginine 15 and isoleucine 16. So that's that first blue square. And then we have two dipeptides that are removed. So there's 2 at 14 and 15, and there's 2 at 147 and 148. So you don't need to know those numbers. I'm just kind of giving you an idea of where they are. But they are completely removed, and when they're removed, it activates chymotrypsin. And then you can see the disulfide bonds are kind of holding those pieces together after the removal of those amino acids. The amount of enzyme available is regulated by limiting its synthesis, and the synthesis of enzymes are regulated by genes. So in gene expression, it will activate the expression of a gene, which leads to an increase of enzyme concentration. So we saw that an increase in enzyme concentration will increase the rate reaction as long as there's enough substrate. But then with gene repression, it will turn off the expression of a gene and lead to a decrease in the enzyme concentration.